Hello, and thank you for joining today's event with Calidi Biotherapeutics, which trades on the NYSE American under the ticker CLDI. With us today, we have Stephen Thiesing, Chief Business Officer, George Peoples, Board Director, Antonio Santidrian, Chief Scientific Officer, and Boris Minev, President of Medical and Scientific Affairs for Calidi Biotherapeutics. We will begin with a brief presentation in a moment, and then we will answer your questions. Users may submit a question at any time by using the Q&A tool at the bottom of the Zoom window. Before we begin, please allow me to read the Safe Harbor Statement. This call may contain forward-looking statements within the meaning of the Private Securities Litigation Reform Act of 1995. All statements pertaining to future financial and/or operating results, along with other statements about the future expectations, beliefs, goals, plans, or prospects expressed by management, constitute forward-looking statements. Any statements that are not historical facts should also be considered forward-looking statements. Of course, forward-looking statements involve risks and uncertainties. I now turn the webinar over to the Calidi team. Please go ahead.
All right. Thank you very much, Craig, and thank you, audience, for joining us this afternoon. You should be seeing systemic and localized anti-tumor virotherapies. That is what Calidi is about. These therapies are designed to attack every tumor and arm the immune system, our tagline being arming the immune system to kill cancer. This webinar is focused on updating investors and potential investors in Calidi's technologies. We will not be covering financial aspects of the company in this webinar. You can find financial updates in our latest filings. And we've already done the forward-looking statements and legal disclaimer. And so now on to the overview. Calidi is a clinical stage company, and we're transforming cancer treatments through innovative oncolytic virotherapies. These are cutting-edge cell-based technologies designed to protect and deliver these virotherapies and ultimately to get them to the tumor site, overcoming clinical challenges of rapid elimination of these virotherapies by the patient's immune system. And you'll learn more about how this works when you hear from the other folks on this call, part of our team. Our systemic, which is an IV-based treatment, and localized technologies are very much about revolutionizing treatment of solid tumors. And with that, I'm very pleased to introduce Dr. George Peoples, who joined our Calidi Board of Directors in June of this year. He is very much a luminary. He's founder and director of the Cancer Vaccine Development Program. He's a professor of surgery at Uniformed Services University. He's also an adjunct professor at the MD Anderson Cancer Center. And we have been working with Dr. Peoples as an advisor to Calidi for the past several years, and very pleased to have him join us as a full board member of the board of directors. I should also add he's founder of LumaBridge, an oncology-focused CRO, and has extensive experience, as you can see, with regard to chair of the cancer care program in the U.S. military. And so he's also a graduate at West Point and Johns Hopkins School of Medicine, and completed surgical training at Harvard's Brigham and Women's Hospital. So I could probably spend 30 minutes talking about his background, but we're very pleased to have George Peoples with us. And with that, George, I'd like you to share a few words. Why have you dedicated time and effort around Calidi, and what's compelling about Calidi?
No, thank you, Stephen. I appreciate that very gracious introduction. And no, we will not spend 30 minutes talking about me. We have much more interesting things to cover. And I do appreciate all of you joining the webinar. And so just very briefly, if you really summarize my career, it's been 30 years of clinical practice taking care of oncology patients and 30 years of working in the IO space, doing some early discovery and development work, and ultimately clinical testing of a variety of new IO platforms. And so what it really has drawn me to Calidi, and I've known the group here for many years, just have recently had the opportunity to join in a contributing role as a board member. But really what has drawn me here, just like my career, it's made up of scientific and clinical aspects. It really is the novel technology that these folks have developed and its potential clinical impact on patients. And so those are really the lenses by which I look at companies and new technologies and opportunities to get involved with are those two items, the novelty of the technology and the potential impact that it can have on clinical patients. I think everybody understands the basic platform here, and that's the oncolytic viral platform. It's been around for a number of years, but like a lot of IO platforms, it's met its challenges and its barriers to really becoming full-blown useful in the clinical scenarios. Obviously, there is an FDA-approved oncolytic viral therapy, so it has gone that far. However, there's still some challenges, and actually, Stephen, in the intro, mentioned a couple of those challenges. You'll hear a little bit more about that. But just like we had the challenge of monoclonal antibodies, it wasn't until they were humanized that they become truly useful clinically. Checkpoint inhibitors, until we understood how to manage the toxicities, that's when we unlock that potential. So what you're going to hear over the next few minutes is the technology that is overcoming those barriers that's truly going to unlock the potential of this oncolytic viral therapy.
All right, thank you, George. We'll move on here. This is a highlight of how Calidi does overcome the obstacles to delivering an effective oncolytic virotherapy. And some folks may have seen this slide previously, but always good to just recap. And that is when we look at these unprotected oncolytic viruses, the challenge being that they are rapidly taken out by the immune system. And therefore, the tumor is left untouched. In Calidi's solution, highlighted here on the right, an allogeneic oncolytic virus-loaded stem cells, and you'll also learn about our new enveloped virotherapy, the RT-Nova or red tail arm. And the key here is being able to protect that OV payload in order to ensure that it gets effectively to the tumor. So the three benefits of the platform that Dr. Boris Menev is going to share with you are supernova or CLD201 is using this stem cell loaded with OV. We incubate over time, get a much larger dose. That dose, the OV, is protected from being taken out by the immune system. And then it also primes, it opens up the tumor microenvironment. That tumor will embrace that stem cell that's been introduced intratumorally. And then within 24 to 36 hours at human body temperature, the OV within will continue to expand until it blows out the walls of the stem cell and then begins to consume the tumor from within. So that's the key, the foundation of Calidi's allogeneic stem cell platforms. And now looking at our oncology pipeline, so CLD-101 Neuronova is in clinical trial right now at City of Hope, and that is moving along quite nicely. We're looking forward to sharing some clinical results in the first half of the new year. And then we also are looking to go into a follow-on, a phase 1b/2 of Neuronova at Northwestern University, anticipated in Q4 of this year. And so looking at supernova, which is what we built the company around, I should mention these Neuronova and supernova have received external validation and funding from CIRM, California Institute of Regenerative Medicine. And then the last on the list here, RT-Nova, which is our newest, most exciting in the preclinical space that we'll share about. And so I'm going to move on looking at the three lead programs here. So Supernova and Neuronova are sister technologies. They both use a stem cell loaded with an OV, an oncolytic virus. The Neuronova is targeting high-grade glioma brain cancers. Supernova now is also an off-the-shelf, and we'll talk about the targeting of advanced solid tumors. And then RT-Nova, our newest addition, targeting lung cancer and metastatic cancers with an EEV, extracellular enveloped vaccinia virus. So with that, I'm going to hand it off to Dr. Boris Manev to tell you a little bit about our Supernova intratumoral administration. And again, this is for head and neck, triple negative breast, and soft tissue sarcomas. Dr. Manev.
All right. Thank you, Steve. Thank you all for joining the call. So I will describe this technology very briefly, but please ask any questions. I'll be happy to answer. So as Steve mentioned, we are planning a new clinical trial to target 3 indications: the triple negative breast cancer, head and neck cancer, and advanced soft tissue sarcoma. And these are all very difficult to treat indications. We are planning intratumoral administration directly in the tumor for several reasons. One, we have really evidence from previous clinical trial with the same virus using patients' own stem cells that we have very solid clinical data, including complete responses. Also, this approach allows very low toxicity, has so-called very high therapeutic index, because all the product that we are using is directly injected in the tumor. And also we documented in previous trials that this leads to very strong activation of the immune system, not only at the injected site, but also systemically. And even we have some evidence of shrinkage or elimination of distant tumors that were not treated previously. So next slide, please. As I mentioned, on the left side is the completed study, which is, again, using exactly the same virus, but it's loaded on patients' own stem cells or autologous stem cells. Now what we are preparing is a much more potent product. In this case, we have very well characterized stem cells that are derived from healthy adult donors. And we spent over 2 years and significant amount of funding to really identify these healthy adult donors and test them for multiple pathogens based on FDA requirements for allogenic donors. So this product has been manufactured already, and we are planning to start the trial in Q1 of next year. Next slide. So this is one of the patients from the previous trial. It is a 70-year-old male with metastatic head and neck squamous cell carcinoma. So this patient was referred to us from his oncologist, who said that he was treated with radiation therapy, with chemotherapy. He didn't respond to this treatment, and basically he said, we cannot offer this patient anything else. So he joined our trial, and he was treated with one single intratumoral injection, but split in 3 portions, and you can see the 3 inflamed areas under day 52 picture. And this is where the injections were done. So again, one single intratumoral injection. And then this was followed by anti-PD-1, which is checkpoint inhibitor, that was applied just one single cycle, and then 1 cycle of radiation therapy. And I have to mention that this patient was treated with the same type of radiation therapy before he joined our trial and did not respond. So we know that in this case, our product, our drug, is very synergistic with both approaches. So, but because of the size of this tumor, it took about 6 months for the tumor to regress completely. Next slide is showing another patient that is a patient with thyroid papillary carcinoma. Again, very advanced metastatic stage 4 tumor. And what you see picture on the left is one single supraclavicular lymph node on the right side of the neck that was treated directly by direct intratumoral injection. And this tumor, as you can see, is very inflamed and red, also very firm on palpation. And it took about 2 months for the tumor to really shrink significantly and also become very soft. And we knew this tumor will regress based on previous studies and data. It took about 3 months for the tumor to regress completely. And even this skin discoloration that you see on the right picture on the right side disappeared 3 months later. Now patient is over 6 years after treatment and is cancer-free. So this patient, interestingly, had also 2 other lesions on the opposite side of the neck that were not treated, but they also became necrotic tissue, and they did not shrink the same way as this one, but they do not require any treatment at this time. Next slide is a summary of our plan for the next clinical trial, which, as I mentioned, will start in Q1 of next year. So we have 3 parts. The first part will be standard 3+3 trial designed for dose escalation. So we have 4 doses and 3 patients per dose. So we expect to have 12 patients. And then only in case we observe some side effects, we are going to recruit more patients in some of these groups. Part 2 is expansion in 3 indications. The 3 indications that I mentioned, we are planning to involve 10 patients from each indication with the dose that was determined from part 1 in this study. So in this case, this is called basket trial to determine the best responding indication. And then in part 3, we have 1 single indication and 1 single dose, but more patients. So up to 50 patients will be treated to really determine the efficacy of our treatment. And next slide.
Thank you, Boris.
Oh, okay. So this was my last slide. Thank you, Steve.
And Dr. Peoples, just wondering if you could share your thoughts on the CLD-201 supernova.
I think primarily, I just want to make sure that all of the listeners understand this is the kind of state of the art for oncolytics, and that is direct delivery into the tumor. And it goes back to some of these barriers that we discussed before about the immune system, the host immune system recognizing the virus and destroying it before it has its ability to do the work. By injecting directly into the tumor, obviously you get the delivery part, but also keeping it within a stem cell allows protection while the virus continues to expand and then eventually can do its work in the tumor in a little bit more of a protected environment. So those are the technological advances that make this viable. But the idea of injecting locally, getting not only destruction of the tumor, but then an immune response generated against those tumor cells, that then that immune system is more systemic acting. And that's the reason why you see responses in tumors that were not injected. It's that systemic immune response against the tumor. Unfortunately, that is not always 100% effective. It's helpful, but trying to inject one tumor and get a response in 100 different tumors is the challenge. And so you've seen in the trial design here that this is after locally advanced tumors, tumors that can be accessed with an intratumoral injection. So while beneficial, and remember the one FDA-approved therapy, TBAC, is also a locally intratumorally delivered therapy. There is benefit here, but where we really would love to go is, can we deliver these oncolytic viruses in a more systemic nature so that they can arrive at the site of all of the tumors, not just the injected tumor. So I think that's what we'll hear about next.
Yep. Thank you, George. Okay, so moving along, we're now going to talk about RT-Nova, which is preclinical. And then this is a technology that was engineered here under the leadership of Dr. Antonio Santidrian, whom you will hear from shortly. And again, this is an extracellular enveloped vaccinia virus. And for those of you that may have been tracking our filings, a couple of weeks ago, we announced the creation of Redtail Biopharma, which is an entity, a subsidiary that right now is wholly owned by Calidi, in order to raise a Series A funding to support and get Redtail, the RT-Nova, into the clinic. And so you're going to hear more about this technology today. We are in active dialogue right now with 5 large cap biopharma companies that have interest in collaboration around this, as well interest in investing in that Series A. And so we're formally kicking that off with a virtual non-deal roadshow next week. But again, Redtail Biopharma, right now a wholly owned subsidiary of Calidi Biotherapeutics. What we're doing here benefits all current and future investors in Calidi. So moving over to Dr. Antonio Santidrian.
Thank you so much, Steve. So before we start the data explanations on the systemic, I want to emphasize a little bit of how important it is also to have an intratumoral. So what Boris has been explained so far is a very powerful platform intratumorally designed for a very specific group of tumors where intratumoral administration makes a lot of sense. And actually, this is why we are so encouraged by the Supernova program. However, it is true that in cancer, we are going to have some stages, some tumor types, where the only way to treat these cancers will be by systemic administration. And this is why we needed to build this platform where the resistance of the virus is so such that now can survive circulation and target those multiple distant tumors. This figure represents the same that was presented before on this presentation, where there is a very limited clinical challenge where anything you inject in circulation, any virotherapy, will be eliminated very quickly. But this new platform, new envelope virotherapy is built to survive that circulation and target all these multiple tumors. Next slide, please. So this platform is also based into using a vaccinia virus. This vaccinia virus, again, is going to be very highly cytolytic and is also designed in a way that is tumor agnostic. So basically, vaccinia virus is not using any receptor or defined receptor, so it's able to infect many, many tumor types, contrary to other viruses or competitors. It's a big virus, it's genetically stable, but also because of that, it allows the insertion of additional payloads in the virus. So we can transform this anti-tumor virotherapy also into a viral vector, able to target every tumor, kill the tumors, but express additional therapeutic proteins. So next slide, please, Steve. Thank you. So how this is built is we are using now a vaccinia virus that is, instead of protected by a whole cell, is now protected by a small cell membrane. It's a cell envelope. No, it's a viral envelope. So we have a new strain of vaccinia viruses that produces very high levels of envelope viruses. So this is important, and that means that the virus is manufactured with this human cell membrane that will contain these human receptors. These receptors will lead the protection and also even the tumor targeting. Now, we are going systemic with this virus, so we need to engineer these viruses to be more tumor selective. It's a totally different safety profile than the intratumoral administration, but this virus has been built on that view. This is also why there is available a safety switch that is a small drug already available, FDA approved, which it will help us to expedite any regulatory pathway and actually the clinical trials. Next slide, please. So again, we build this new virus, which has this capacity to be manufactured in very high levels of this with this extra envelope. So what that means? So in terms of what happens with the virus when the virus reaches the tumor is what this picture shows. Let me show you first on the left side. That's a prior generation of a vaccinia virus. That virus does not produce these high levels of envelope virus. They are secreted. So for that reason, this virus will remain localized in the tumor. So when one particle reaches the tumor, it will spread locally and infect the neighbor cells. And this is good, it's efficient, but we can do better. So now the new virus that has this new characteristic of being secreted with an extra envelope. What that means visually is that when the virus infects a tumor cell, now the virus is able to be secreted and jump very far away. And you see here these tails, in red, is actually the virus in red that is able to infect the cells in forms of a tail. That is the origin of the name of this virus that we call red tail, and mimicking the red tail hawk and visually a red tail. Next slide. So now we have that virus. It's very powerful, tumor selective, able to be protected. But now the important is we need to be able to manufacture that. We need to keep during the manufacturing process the stability of that second membrane. And this is why our team has been specializing for many, many years. It's not only we have a very cool or very good technology, we know how to manufacture them and go to clinical trials and commercial. These pictures is a very a beautiful experiment where on the top, we are doing an experiment to quantify how many viral particles we have. We are using a virus that doesn't have this envelope. And when the virus is incubated with human serum, you see on the right top how the virus is killed very quickly. However, when we have a virus with this extra envelope manufactured using our protocols that keeps the stability of that membrane, now you can see how the virus is able to survive human serum, then human blood circulation. So on the next slide, now that we have the virus, we know how to manufacture it. We can do now very beautiful experiments where the virus is injected systemically and then we can analyze how it targets multiple tumors. This animal experiment, we created a mouse that we can create with multiple tumor types. In this case, head and neck, lung cancer, and melanoma. So you can see on the right side how the virus is labeled with a color, is able to target every single one of the tumor sites, even if there are different tumor types with different tumor microenvironments, with a very good efficacy of tumor growth inhibition. This is an impressive experiment. Obviously, these mice, if you can recognize them, they are immune deficient because here we are using human tumors. However, we have a lot of data of animals with immune competent background. For example, this slide, this animal has a lung tumor, mouse lung tumor, in a model that is full immune competent with full immune system. The tumor was implanted subcutaneously for easy visualization, and the virus was injected again systemic in the tail vein. You can see with the picture on the left how the virus targets very specifically that tumor, and the picture in the middle how the tumor, the control tumor in black continues growing, while the treated tumor, we have a very, very remarkable response, even with a very low dose of 10 to the 5 particles. This is impressive data on a virus able to survive circulation and target distant tumors. But we are not still satisfied with that data. We want to go further. We wanted to analyze if this virus is able to target multiple metastases, not one tumor, not 3, multiple, 15, 20. So this is why we created these models where we build animal models with multiple metastases. In these pictures, there are pictures of the organs of the animals. On the left, there are the lungs of animals. They were implanted with lung tumors. You can see on the top picture where the mouse is, the color of these tumor cells in multiple locations, in one animal or two animals. On the number 3 and 4, this is the lungs of the lungs of the animals, they were treated with this systemic virus. On the bottom, you can see in red how this virus is able to target the same locations of these metastases in the lungs. On the right side, you can see different organs. Those are the livers of the mouse. There where the mouse is are the control animals with the liver metastases on these blue, purple colors. On the right side, you're going to see the treated animals. And this 6 days after the treatment, it was almost very difficult to detect any metastases on these situations, indicating the high efficacy of these treatments. We want to target multiple tumors. We want to kill them. But what is remarkable is that we want to change the tumor microenvironment of every single tumor, not one, not two, every single one. On these pictures, we have histopathology of lung metastasis. On the left top, you're going to have H&Es where you can very easily identify those nodules of tumor metastasis. You can count very condensed blue, more than 15 loci. On the immunofluorescence pictures on the right, you can see how we can zoom in and we almost cannot detect any infiltration of immune cell. On the bottom is another animal with tumors animals that were treated with the vaccinia virus. We can go and focus right now on the right side. You can see in yellow the virus targeting every tumor, and you can see the massive infiltration of tumors of immune cells in light blue CD8s or in pink CD4s in every single tumor or metastasis that the lung had. So this is again impressive data that was presented originally in ASCO meeting just recently, which really attracted the attention of many researchers and people. So with that, as a conclusion, we have a very good plan that has been discussed with regulatory, actually also here with George people. We are comfortable to say that our first indication for this treatment is going to be metastatic non-small cell lung cancer. Is a type of tumors where it's a very high unmet need, where the survival is median survival is around 6 to 12 months, and the survival rate of 5 years is less than 8%. We are going to be focusing on a phase 1 trial in around by middle of 26 to start there with a design of a regulatory path that allow us to request the FDA an expedite path. And because we are focusing on an unmet need, we believe that our design is going to be a phase 2 pivotal trial where we can potentially have a regulatory approval targeting late 2029. As everybody can see, we are designing these trials to have as soon as possible proof of concept, but also as soon as possible a drug that can go commercial in this type of cancer. So with that, I would like to complete the presentation. We have a summary of the slides, but I think we're going to stop here and Steve will take it from here.
Yes, just unmuted. So thank you, Dr. Santidrian, and Dr. George Peoples, if you would maybe just share a few thoughts on the RT-Nova technology and Redtail Bio before we go into audience questions.
And I'll keep this short in the interest of time and allowing some time for questions. But I think everyone who's listening has a pretty good sense of the overlapping nature of these two technologies. The direct intratumoral delivery of supernova is ready for the clinic. Now it has already been tested in its first clinical trial You saw some of the clinical responses there. It's ready now in its allogeneic form to go into those tumors that are accessible to intratumoral injection and in patients that have limited metastatic disease. So there's a clear-cut path for supernova, and that path has been delineated. The exciting news around RT-Nova, obviously, is this has been somewhat the holy grail of oncolytic therapies of how can you deliver an oncolytic systemically protected, get it to the site of tumors, and allow it to do its work. And I hope you're equally excited as I am about the technology that Antonio has described that is going to get us to that realization here in the very near future. So I'll pause there. And Stephen, I think you're going to open it up for questions.
Yes. So Craig has actually been gathering questions from the audience. And
yes,
audience, you're welcome to submit questions. So I'll hand it over for the questions, and then our team will respond.
Thank you very much, Stephen. First question is, how do you assess the long-term efficacy and potential side effects of your therapies in clinical trials?
Yeah, I can answer here. In our clinical trial design, we have specific time frame for to analyze the toxicity first, and this is about 1 month. And why 1 month? Because we have seen in the past that most of the potential side effects are just the local inflammation and redness in the injected site, which last for 1 to 2 weeks. But we are going to monitor, and this has been reviewed by FDA as well, for a month specifically for toxicity. For efficacy, we are going to follow the patient for 1 year. And to determine the efficacy, we are using so-called RECIST criteria and also immune-related RECIST criteria, which are specific measurements of the tumor and evidence for tumor shrinkage that was done based on imaging every 3 months, like month 3, 6, 9, and 12. And then we are going to determine in this way how efficacious our treatment is.
And
Thank you very much.
The only thing I would add to that is, do recall the autologous version of Supernova has been tested, and those patients have been followed for multiple years. So there is already some existing long-term data, both safety and efficacy.
All right. If we're finished, Calidi, we can move on to the next question.
Please.
Yes. Okay. And I just wanted to remind our participants, thank you very much. We have received a cascade of questions today. I would like to ask that anyone who wants to ensure that their question gets to Calidi, because we may not be able to get to all of our questions today, please email us your question at cldi@redchip.com, cldi@redchip.com. That way we will ensure that the Calidi team will see your question and you will receive an answer through email. Next question is, what are some challenges in delivering virotherapies to tumors?
Well, if I can start to answer that, obviously the first challenge is the quick elimination by the immune system. That's why Calidi is here, not to actually bypass that challenge by protecting that virus and reach those tumors. Obviously, there are other challenges. There are the challenges that every single biotech has. It takes time to develop these technologies, but we are here. We have the right team, the right expertise, and we are moving full speed. If anybody else wants to add, Maurice?
The other thing I just wanted to add is related to what you just said. It is a modification of the tumor microenvironment in a way to support the viral replication and killing of more tumor cells. And this is what our stem cell-based technology is doing, because the stem cells not only protect the virus, but they also amplify the virus, and also they modify the tumor microenvironment. because they secrete some specific growth factors and so-called cytokines or chemokines at the injected tumor site to allow the virus to kill more efficiently tumor cells.
Thank you.
Next question. Tell me more about the safety profile of your treatments.
So again, first, we are using vaccinia virus, which has been already approved by FDA as a safe virus, as a vaccine for smallpox. So we know that this virus is not a human pathogen, so this virus cannot cause disease in patients normally. But also, we really have significant experience with this virus in previous clinical study that was done with patients' own stem cells. And so both of these sources of information are telling us this virus is very safe. Again, we will have significant precautions in the trial that we are initiating because they are available, 2 antidotes for this virus. We'll have them available in the clinic just in case we observe some side effects, which we don't expect, but it's good to be prepared for these cases.
And just to
Next
add,
question.
just add briefly, the safety study that Dr. Manav refers to, you saw 2 patient images from that safety study using the vaccinia loaded on the payload of stem cell, and that was 26 patients. So you saw only 2 of 26. Safety was shown in all 26 of those patients. There were no significant side effects. Go ahead, Craig.
Yes. When do you anticipate getting RT-Nova into human clinical trials?
Yeah, maybe I went too quick on that slide. No, the idea is to be in clinical trials by 2026.
Thank you.
I'm sure most of the listeners are aware, but once you go, with a new technology, there is a process that has to be followed with the FDA to get them comfortable with the idea of it being sufficiently safe to put it into patients. And that process goes through what was on the slide, the INTERACT, the pre-IND, IND process, and that takes the better part of a year and a half to complete to get to that first human study.
Thank you, Jordan.
And gentlemen, I think we have time for one final question. When can we expect to see clinical updates on Supernova?
So again, we are planning to start the trial around February, March of next year. And the first readout for the safety for the first injected patients will be a month later. But again, we need to recruit 12 patients and so to monitor the safety. In terms of efficacy, we need about 1 year to really have a better idea on the efficacy because we need to do, as I mentioned, multiple imaging studies and to analyze this data.
And just to kind of add on to that, just as I was mentioning with the process, even though there's been a trial already conducted with the autologous version of this vaccine, or excuse me, this virus, this allogeneic delivery system is new and therefore has to go through the FDA's IND process also. And so all the work has been done. That IND is to be submitted before the end of the year with the start of the trial, hopefully by the beginning or the end of first quarter of next year. So that's the rough time frame on getting the Supernova study started.
And I just want to add one more sentence. So we expect significantly better efficacy in the new product because of 3 reasons. One, this is really very well characterized product. We know that we have 100% stem cells that we need. The second is that in each stem cell now we have many more viral particles than in the previous trial. And so this is really very important. And the last thing is that now we are injecting the patient's tumor 3 times, not just once as before. So this is significantly more promising strategy and more potent product.
Thank you.
And thank you, everyone, for your questions and to the Calidi team. Let me just give that email address one more time. The Calidi team definitely wants to hear your question, and it will be easy to submit it by using CLDI@redchip.com. CLDI@redchip.com. Additionally, we have an investor information page just for Calidi. It is CLDIinfo.com. There you can view and download the investor presentation and fact sheet, as well as sign up for news alerts for Calidi. That is it. Thank you very much, gentlemen, for your presentation today, and thank you to our many participants.
Thank you.